Good morning, everybody. We're going to wait for more participants to trickle in before we start the webinar today. Okay, we're going to go ahead and get started, and we're going to start with a few announcements. So welcome to the RCN for wastewater surveillance for SARS-CoV-2 and emerging health threats. Our webinar series has one webinar per month, the first Friday of the month, so welcome. Today we have two sets of speakers, so we're really excited to have Audrey Wang and Minxi Jiang from UC Berkeley. They are PhD students who will be co-presenting their presentation. And then we have Auke Haver from RIVM, National Institute for Public Health and the Environment in the Netherlands, who's also a PhD student, who will be talking about his work at Schiphol Airport for wastewater surveillance. So really excited to hear the talks. Thank you for joining. Before we begin, we want to remind you that we have three activities to be aware of in this slide. One is the monthly journal club, and the next meeting for that is November 13th. And we also have nominations for the webinar if you would like to nominate someone for speaking at the webinar this academic year. And then we have an email list, and Sarah is putting some links in the chat for you if you're interested in accessing these resources and you currently don't have access to them. And then lastly, Sarah wants to make an announcement about this workshop.
Yes, our next workshop is on December 5th from 6 to 8 a.m. Pacific time. We're focusing on WBE and lower and middle income countries. And we want to talk about the logistics of getting a sampling program set up, and how to develop actionable programs and what that would actually look like. We have a really good panel set up. This is going to be a little bit of a smaller workshop, so everyone is allowed and welcome to submit an application, and then we will meet to decide who joins. We want to make sure that it's a pretty focused and smaller conversation rather than one of our larger ones, and I'm putting that application link in the chat now. And we're all set.
Thank you, Sarah. Okay, so while Audrey and Minxi share their screen, can you guys go ahead and share your screen? I'll just explain for those of you who might be new how the webinar works. Each set of speakers will speak for 20 minutes, and then there'll be 10 minutes of Q&A, which will be moderated Q&A. And if you have questions in the audience, you can put your questions in the Q&A in the Zoom, and the moderator will ask them, and you can upvote questions that you see that you'd like to be asked. So with that, I will pass it off to our first speakers. Thank you. Minxi, you guys are muted, just so you know. Okay.
All right. Okay. Can you guys hear us?
Yes, we can hear you.
Awesome. All right. Thanks for coming to the webinar. It's our pleasure to present. My name is Audrey, and I'm currently a PhD student in Nelson Lab. And Minxi, she's a postdoc, also in Nelson Lab. And today we're presenting the topic, benchmarking concentration extraction methods for human viruses from wastewater. And I will cover the first part of the talk, including background and the TPCR spike-in experiments, and Minxi will talk about the sequencing in the later part. All right. So one of the main motivations for our study is that our targeted human viruses are often at low concentration compared to the bulk microbial wastewater background. And this challenge has led to the development of different concentration and extraction methods for wastewater samples. And here are some examples for the well-established methods, including but not limited to site selection-based ultrafiltration, electronegative membrane, and pack precipitation, and etc. However, these methods have limitations that they require large processing volume and which will take longer processing time. So when the pandemic started, there's a need for more efficient methods that allows a smaller effective volume. So here are the emerging methods since the pandemic, and we selected them in our study. And because these emerging methods has been monitoring mostly on SARS-CoV-2, so another motivation for us is to be able to go beyond SARS-CoV-2 and monitor a wider range of viruses for the post-pandemic time. And as we know, different viruses have diverse virus characteristics, forms, and emerging variants. So this also motivates us to understand the method-specific limitations and biases of detecting different human viruses. Here's the central goal of our research is to benchmark different concentration methods for human viruses detection from the wastewater. And for our study, we have two endpoints detection tools. One is dPCR for the spike in wastewater, and the other is the sequencing for the unspiked wastewater. And the objective for the dPCR is to understand the sensitivity of methods for a range of specific viruses. And for the objective of the sequencing is to study the effects of methods on detection of a broad range of human viruses. So these are the 4 methods selected in our study. The first 2 methods, Promega solids, they are a direct extraction method, and InnovaPrep and NanoTrap are concentration methods, which will be followed by an extraction kit. And in order to standardize these 4 protocols, we use the same extraction kits, the power viral kits. And we also have the starting material as the 40 ml effluent wastewater. And they all ended up in 100 microliter elute total nucleic acids. So let's get into a closer look of all these methods. Bear with me. The first one is the Promega method. It starts by adding protein A's, and we follow by a partial solid removal, and the whole supernatant go through the silica column-based tube for direct extraction. And for the solids method, the samples go through the high-speed centrifugation, and the solid pellets are collected, 0.25 gram, go to the power viral extraction kit with the bead beating step. And for the InnovaPrep method, it's a concentration method for the liquid part of wastewater. We first added 2020 to detach the virus from the solids. And then we do the solid removal followed by the ultrafiltration. And the concentrate further go through the extraction kit. And for NanoTrap method, it is a concentration method on the total wastewater by adding the affinity-based magnetic beads, and then we apply a magnet to concentrate out all the viruses, and the concentrate also go through the extraction kit. And these are the 2 endpoints methods we use. One is digital PCR that can have the sensitive and specific quantification of virus targets, and sequencing method that genotype multiple known and novel viruses. And below are the common 3 wastewater sequencing methods, including metagenomics, targeted amplicon sequencing, and probe capture enrichment targeted sequencing. And for our study, we selected the probe capture enrichment target sequencing, and Minxi will talk more about it in her part. So now I will talk about the dPCR part and how we benchmark methods by a range of spike-in human viruses. So these are the spike-in virus targets we selected in our study, including coronavirus, influenza, adeno, and enterovirus. And for each virus family, we selected two strains so that we can test the trends across methods and to see if they are consistent within each virus family. And here you can see that we selected the virus that can cover a range of physical and chemical properties. There are envelope and non-envelope virus, and RNA and DNA virus. And all these viruses, except for SARS-CoV-2, are cultured by our collaborators in Lawrence Livermore National Lab. And SARS-CoV-2 was requested from BEI. So here's the overall experimental design for the spike-in experiment. We collected samples at 3 wastewater treatment plants with different scales, small, medium, large. And we collected at 2 time points, 1 month apart. And we tested on 4 methods in triplicate. And there are 3 unspiked background controls. And for the virus cocktail preparation, we mix all the virus culture by L and L into this virus cocktail, and then further spike into each method in triplicate. And then when we incubate on the rotor to let the sample to reach homogenize and equilibrium. On the next day, we perform the 4 methods in parallel, and here's the collected total nucleic acids and go through the downstream dPCR quantification. And all the assays are designed manually by Rose, and she's the PI of our project. And the assays are further validated and optimized in the lab. So before I jump into the result, here's the back calculation of our virus concentration. So here is the concentration detected on dPCR, and we did some back calculation and get the total gene copies in 40 ml wastewater. And this indicates how much virus recovered from each method. And for my next slide, I will report the log 10 of the total gene copies in 40 ml wastewater. So here you can see that for each box are different methods, and y-axis is the log 10 total gene copies in 40 ml wastewater. And here this is the targeted spike in gene copies. We aim 10 to the 6. And here you can see that Promega and nanotrap gives a high recovery for coronavirus, and it's quite consistent for these two with these 2 strains. And now let's look at the rest of the viruses. And here you can also observe that Promega gives a great recovery for also influenza and adenovirus. However, for Kawasaki virus, it shows a different trend, behaves differently, that the solids and nanotrap recovers higher for A6, and they perform differently even within the same family. Another thing to highlight is that the overall recovery of influenza is relatively lower than the other viruses. And we have the evidence, and this is not shown in this presentation due to the time limitation. So we have the evidence suggested that there's a high proportion of influenza in extracellular form, so it's more susceptible to degradation. And for the next slide, I will report the result in TNA concentration. So this is the total nucleic acids that's collected after each concentration extraction method. And this is the material that will be put directly into the dPCR machine. And this can help us to check the detection limit of the dPCR for each method. So here you can see, sorry for the small font, but x-axis are different methods, InnovaPrep, nanotrap, Promega, solids. And for each plot is different virus. And you can see that there are some really low TNA concentration. And this is just for us to bear in mind when we design the experiment. Like if here, this is the given starting gene copies of 10 to the 6, we need to be aware of the dPCR detection limit of especially IP and solids method. And I will pass the slides to Minxi for the sequencing.
Hi, everyone. My name is Minxi. I'm the postdoc in Nielsen Lab. So for my part, we use an unspiked wastewater, and our goal is to see the impact of those concentration methods on the target sequencing of human viruses from those unspiked wastewater. Well, here's an overview of our experiment design. For the sample preparation, we collected the influent wastewater from 1 wastewater treatment plants. We have 3 sampling dates, and we applied the 4 methods Audrey already introduced to extract the total nucleic acids. And all the extracted nucleic acid went through the quality check, including the qubit and also the fragment analyzers. The qualified total nucleic acid was divided into 2 parts. One part went through the dPCR quantification, another part prepared for sequencing. For library preparation, we have selected the probe capture enrichment panel from Illumina. So this panel targeted 66 groups, around 200 strings, virus genomes, and they designed the probe to hybridize to the target genomes. Because of the hybridization, which allows more mismatch, we could have some rooms to detect both known and novel viruses. And after the hybridization, those bonded targets will be separated from those unbonded DNAs because they have the biotinated baits. Following the cleanup and amplification, like the prepared library will be loaded to the sequencer, which is NovaSeq in our research. And finally, the resulted raw sequence was analyzed by customized bioinformatic pipelines. Basically, this pipeline includes both the read-based classification, which allow us to see the different taxonomy domains and identify human virus through a host screen. On the other side, we also include the assembling-based analysis, which allow us to recover near complete genomes and can go through the following genotyping or the phenogenic analysis. Well, here we first show the results of the quality of the extracted total nucleic acids. On the left side, you can see the results of the qubit. It's clearly that solid will give us the highest yield for both DNA and RNAs. And we noticed that for all the samples, higher RNA concentration was extracted compared to the DNA. On the right side, we show the results of the fragment analyzer of the RNAs. And you can notice that there is a very distinct size distribution from the NT samples. Especially, there's no peak for 16S and 23S. So from those raw results, almost like many of the human virus were RNA virus. So from those raw results, we wondered that whether those high RNA concentration in solid samples and less RNA in NT samples will allow us to detect more human viruses. Well, then we show the results. First is a read-based classification of the taxonomy domains. We plotted the graph with a percentage of those reads and divided by the total unique reads. As you can see from this graph, not very exciting, but almost all the methods, they have a lot of reads assigned to bacteria or unclassified. Especially NT samples, we notice that there's a majority of unclassified reads. This indicated that probably some novel virus was captured by the nanotrap beads, which cannot be classified by the current classification tools, or just due to the limitation of the virus database. Of the database, not virus database, sorry. Well, then we zoom in to the virus proportions, and here we show the results. Well, it is very clear to notice that instead of NT and instead of a nanotrap and solid samples, IP, which represent the Innova prepare concentration pipette, they showed the most sensitive detection of the total virus and also the human virus. And we further look into the virus host distributions of all the classified viruses. And we noticed that besides the human virus, like the most of the classified virus was assigned to bacteria host in nanotrap and the solid samples. This means most of the classified virus in the NT and solid samples are actually bacteriophage. So we're trying to look back for our method and see what will cause this difference. And we noticed that for both Promega and IP methods, they actually include a solid removal steps after adding the proteinase and 2020. However, for solid and nanotrap methods, they just include the total wastewater. So this gives us the conclusion that IP and Promega methods might strike a balance between eliminating the solid associated bacteria and bacteriophages while still attempting to retain the human viruses. Lastly, we also compare the detection sensitivity of endogenous SARS-CoV-2 by dPCR and targeted sequencing. So from this graph, you could see that the blue bar corresponding to the left axis is a quantified concentration by dPCR. And the red dots, those red dots are corresponding to the right side, and that's classified SARS-CoV-2 rates from the sequencing. So from this graph, we noticed that although IP methods yielded a very high percentage of overall human viruses, but it yielded a negative dPCR quantification in samples that actually tested positive via the NT and Promega methods. And the Promega methods, we noticed that it's consistently and very sensitively detected the SARS-CoV-2, which quite corresponding to Audrey's spiking results. Due to the time limitation, we cannot show more results of the sequencing, and we'll come to the conclusion for today's presentation. Well, basically, we have selected the 4 methods that is widely used during the pandemic, and we have 2 endpoints of virus detection, including the dPCR and probe capture enrichment target sequencing. And under our experiment conditions, we noticed that for those spiking viruses, including coronavirus, influenza A, and adenovirus, Promega might be the best method to detect them. However, for the overall human virus, IP method might be more sensitive compared to others. This highlighted the importance to align our wet lab and endpoint detection of different viruses based on the research goals. There are some other aspects we didn't cover today, such as the human virus diversity, and we also have the assembly-based virus subtyping results. We're also doing the LOD testing, inhibition testing, and different virus forms analysis. That's all of our presentation, and here is our contact information. We want to express our huge thanks to the amazing research groups at UC Berkeley, our PI, Kara, Rose, and other colleagues, and also our collaborators. And we are welcome for any questions. Thank you.
Great. Thank you, Audrey and Minxi. And a reminder for all the attendees, if you have questions, you can add them in the Q&A tab. So the first question that we have is, did you see any major differences between your small, medium, large wastewater treatment plants that you sampled from? And I believe this was for Audrey in the first part of the presentation.
Yeah, so currently we're trying to do some like regression analysis because we collected the metadata from different wastewater treatment plants at different time points. And we do see that the population flow rate and TSS BOD values vary across wastewater treatment plants. So we tried and we did a PCA analysis and seeing that they cluster distinctly according to the wastewater treatment plants. However, we haven't really dig into like what are the contributors to this distinct clusters. But we do see a difference.
Interesting. So the next question we have is, do you have any plans to test solid removal prior to NT as well, or also to look for non-viral biomarkers?
Yeah, so for the spiking experiments, we did not do a prior solid removal for nanotrap method. And I believe for sequencing, there are more
for sequencing samples, we didn't do like the solid removal. We just include the wastewater.
and other targets?
Sorry, what's the other question? Can you repeat?
Non-viral targets.
Non-viral targets, you mean like bacteria?
Yeah, or fungi.
Oh, yeah. So for the sequencing, we do see that a lot of rates was assigned to bacteria, but we didn't take a deep look into what bacteria was there.
Okay. Great. Did you see any challenges with the sequencing depth using the probe-based enrichment that you used?
Yeah, that's a good question. So we have like around 15 million reads for each samples. And under that depth, we see like around, so IP samples have around up to 2% of the reads assigned to viruses. So I guess that's just a reference for other people who want to do the probe capture from Illumina. Yeah, that's what we got.
Okay, great. Did you account for dimers that may be quantified in the qubit or see any dimer effects downstream?
I'm not sure about that.
Sorry, like you mean dimer quantified by qubit?
Or is it
I'll be honest with you, I'm not familiar with this either, so I can't give any more insight into the question. But perhaps when we're done with questions, you can go into the Q&A and maybe have a back and forth with Dagmara, who's asking this, and we could move on for clarification. So Marlene asked, when we work with solids, they've found that direct extraction didn't work as well as some other approaches. Did you try any other approaches, such as use of buffers before extraction or anything else?
Buffer, you mean like what kind of buffer? So for the direct extraction from we directly use a kit from Promega. And I don't think they have any buffer adding before extraction, but yeah, I'm not sure what you refer to as a buffer. Do you mean like the RNA DNA protector or something?
Perhaps a lysis buffer, but I'm putting words into
so Promega. Yeah, so Promega methods, they have the step to adding the proteinase before the extraction, before the solid removal. Yes. Oh, okay. Yeah. Before the solid removal. And according to their paper, so the proteinase is not acting as a lysis buffer. It's just a proteolytic like cleavage steps to try to release the virus from the solids.
Got it. So one of the panelists, Ali, asked, did you account for inhibition and I believe she asked this earlier in the talk, so I'm guessing this is for the spike-in study.
Yes, we did do a 1x, 5x dilution and to see if there is like inhibition happening. And we did observe from the Promega, it shows a relatively higher inhibition compared to other methods, but also different methods perform differently. So we still need to further confirm that. Different viruses perform differently, so we need to further confirm.
Great. And I think we have time for one last questions from Sarah, one of the panelists. What was the coverage quality of the sequence reads? Could this affected the unknown assignments?
Yeah, that's a very good question. So the coverage, we actually, I have the data, but I still need to, I didn't remember the numbers here, but I can look into that and reply you like later. Yeah, we do recover some like near complete genomes, but it's not that much numbers. Yeah.
Great. Well, thank you, Audrey and Minxi. I think we're going to welcome our second or third speaker, our second talk for today.
Okay, great.
Thank you.
Thank you.
Great. So while all the slides are getting pulled up, I'm excited to announce that our second speaker is Auke Haver from RIVM, who's a PhD student. So whenever you're ready, you can go ahead and get started.
Okay, well, thanks for having me. My name is Auke Haver. I'm a PhD student by foundation at the Wastewater Surveillance Department at the Dutch Institute of Public Health and the Environment. And I'd like to talk today about the work we've recently submitted, and we've put a preprint online. Well, before I talk about the research specifically, I'd like to talk a bit on a personal note. Before I worked at the RIVM, I studied for my bachelor's and master's, my undergraduate and graduate degree at the VU University in Amsterdam. And if you've studied at the VU University in Amsterdam as I have, it's likely that you've lived at the student campus in Ouderstede, which is where this photo of me is taken. I still live there. And one thing that's associated with this student campus is airplanes. And that is because the student campus is along one of the approaches to Amsterdam Schiphol Airport. And I have felt this the past 8 years at least, because there are times when you have an airplane going over your head once a minute. And that's because Amsterdam Schiphol Airport, or AMS, which I'm going to be calling it, is one of the busiest airports by international travel worldwide. So pre-pandemic, it had about 71 million passengers moving through it in a single year. And because of these huge numbers of passengers moving through this area on transit to different continents, to different parts of Europe, it became one of the first locations of interest for sampling of SARS-CoV-2 viral loads to really track this ongoing pandemic. So the first case in the Netherlands was detected on February 27th, 2020. Now, ahead of that, there was already sampling at the wastewater treatment plant going on. At that time, there were two groups working on it, our group at the RIVM and the group at KWR, led by Gert-Jan Medema. And what both groups observed was that they did not detect a positive signal for SARS-CoV-2 ahead of this first case, but soon after this first case was detected, there was a positive signal. Now, we at the RIVM have been sampling at this location and at every other wastewater treatment plant in the Netherlands since the start of the pandemic, and we still are. But I'd like to focus now on Amsterdam Schiphol Airport. When Anne Merel and I started our PhDs in 2022, Anne Merel van der Dift, our first project together as PhDs was looking at this data we had collected, or that has been collected at Schiphol Airport. So sampling had been going on for 32 months. In that period, 277 samples had been collected and analyzed to detect viral loads. Now, this sampling had started with weekly samples, and that had been scaled up to a level of 4 samples per week. And now, out of those samples, 177 samples had been sequenced, so we could track variants. Now, specifically, the sampling had been performed at the wastewater treatment plant, which services the terminals, the airplanes coming and going, and around 70 companies that are located on the area of the airport. But it does not process rainwater, and it does not process household wastewater. So it's a very good view of what's going on at the airport specifically. And our samples were all flow proportional influent samples taken in a period of 24 hours. So when we started looking at this data and looking at the existing literature, we saw that there had been a lot of interesting studies performed on travel hubs, on long-term surveillance at travel hubs, at using sequencing to detect incoming variants. But we hadn't really seen a lot of studies that combine these elements, that perform sequencing on and monitoring on really large timescales at travel hubs. So we wanted to combine these aspects. So our approach was to look at what was happening at the time, what were control measures that could have influenced the number of passengers, at Schiphol Airport, so lockdowns, flight cancellations, mandatory face masking. We wanted to see what were those passenger counts, what were the pre-pandemic numbers, what were the numbers during the pandemic. Of course, we had the viral load data from Schiphol, but because we also do sampling at every other wastewater treatment plant in the Netherlands, we had this viral load data for 99% of the Dutch population. And for variant data, we had performed sequencing, and we wanted to compare those estimates we could get from wastewater to what was being seen in clinical reporting using test samples. So I'd like to take you through some of the results that we observed. And I'm going to focus most on variant data because I'm a bioinformatician and that's where my passion is, but I'm also going to show all the other parts. So one thing we saw was, of course, at the start of the pandemic, passenger numbers, of course, through measures, plummeted. Where we had 100,000 arrivals per day, we dropped to a low of 4,000. And what we saw was, at times, it slowly recovered, but it took well into 2022 until the number of flights, number of people passing through Schiphol was back at the same level. However, even while we had these really low passenger numbers, we had very little no detects at Schiphol Airport. So we had 4 samples which did not have a positive signal for SARS-CoV-2. 2 of those, as I've previously mentioned, had been taken ahead of the first clinical case being detected in the Netherlands, and 2 were detected when passenger numbers were relatively low compared to what we had seen before the pandemic. But if we look at the samples taken at the 24th of May and the 7th of July, they did not occur, and I've shown them here in yellow, they did not occur at the absolute lowest number of passengers. And at similar moments, we were able to detect the virus. Now, we corrected these viral loads for the total flow because we had, of course, these numbers. And what we saw was that the flow correlated really well with the number of arrivals. And something that was really interesting was when we used these flow-corrected viral loads, we saw actually that in June 2021, around the time of the start of the Delta variant, and here in May of 2022, which was around the period that the BA.5 variant was introduced, we saw these huge general increases in viral load. So I've changed the axis here a little bit. It shifted. These axes are 5 times as high as what we observed here, and these are 10 times as high as what we observed here. So somehow there was proportionally more virus material being shed per total passenger. So maybe more passengers were shedding into the wastewater. Of course, we wanted to look at these trends and compare them to the national figure. So we compared them to the national viral load, which we could correct for, of course, flow, but also inhabitants, because we know who is connected to which watershed. And what we actually saw is, and I've shown this in pink, when there were moments of peaks in this national figure of viral loads, we see this similar peak at Schiphol Airport. And sometimes we even see these peaks occurring a little ahead of what we saw nationally. And that makes it, of course, a very interesting location to sample. Now, of course, I'd also like to talk about what we did with sequencing. Of course, we performed sequencing to detect the different variants and see what is the diversity of the virus. We did this through sequencing of the cDNA amplicons. So we used the Arctic V3 and V4 eventual primer schemes. We sequenced paired end on the Illumina MySec platform, which generates 300 base pair reads. For each sample, we have between 1 and 3 replicates. Now, of course, we did some cleanup of these reads, and we used the Freya method to estimate the relative abundance of different lineages. Now, this is performed. I'll shortly explain how this works. Of course, knowing the phylogenetic tree of SARS-CoV-2, we're able to associate substitutions to different lineages. Using the Freya method, we obtain a matrix with substitutions and lineages, and it's a binary matrix with association. Using that and the substitutions or SNPs we observe in our sample, we're able to fit a weighted least absolute deviation model where we say, well, the minimum frequency of lineage to be reported is 0.1%, and the sum of all frequencies need to be 1. And from that, we're able to get a really nice estimate for the variant composition in our sample. So this is an example of how such an estimate looks like. And these, of course, are all delta lineages. So we performed this for our wastewater samples. So we had 177 samples sequenced, 161 were of sufficient quality, which meant we had at least 50% genome coverage with a depth of at least 10 reads. And these did not have demixing errors during this lineage abundance estimation model. And we compare that to the RIVM clinical open data. So in the Netherlands, we have the chemsurveillance program, which tests patient samples and test swabs. And during this period, around 130,000 samples had been taken and sequenced, which is about 1,000 to 2,000 per week, and which is reported as a weekly figure of variant of concern, variant of interest, and variant under monitoring counts. And now, of course, these two data types are slightly different. So our approach was to further bin these lineages and variants into the relevant variants of concern and variants of interest. So, for example, the alpha, delta, omicron sublineages. Now, for those really familiar with the SARS-CoV-2 phylogeny, we did detect the beta and the gamma, or the chemsurveillance did detect the beta and the gamma subvariants in Dutch clinical samples, but they occurred at very low frequencies, and in wastewater we only detected these at very low frequencies. And the other variants under monitoring, they weren't detected at all, even. So looking at the results of this comparison, first, just looking at wastewater, so our highest sample density was between November 2021 and September 2022 for sequencing. So I'll focus on this period. And this period contains the Delta, BA1, BA2, BA4, and BA5 variants. Now I've put these variants here as bars, showing a daily measurement. And what we see actually is when we have this replacement of the Delta lineage by the BA1, we see this increase in viral loads. And similarly, when we have this introduction of the BA2, quite rapidly, we have, again, a slight increase in viral loads. It's most pronounced, in fact, when we had the introduction of the BA5 variant, and we see this clear increase in viral loads, which was also previously observable in a different plot. So there is a clear pattern of viral load increase as a variant emerges. Now, we also want to compare, of course, this wastewater data to the clinical data. So what I've done is I've overlaid these daily wastewater samples onto the weekly estimates of clinical sampling, which is visible here. So here the clinical sampling is a background, and the wastewater samples are on top. And what we see actually is a very similar pattern of variant emergence compared to when we compare the sampling at Amsterdam Schiphol Airport to the national clinical surveillance. What we also detected was an Omicron BA4 variant at very low frequency, even 2 months ahead of clinical detection. However, this is around the time of the BA4 emergence worldwide. So this would need further validation. We also detected some we also detected some recombinants. And of course, currently we're in a period where there are mainly recombinants circulating. At this time, when we look at December 2021, 2022, there were very few recombinants. In fact, in clinical surveillance, they're very very few recombinant lineages, and we detected quite a lot more. Luckily, these are all recombinants of the then circulating lineages, so Delta and Omicron recombinants. And what we see here, these are all BA2 and BA5 recombinants. So this could be just a feature of our method used. So summarizing this, we were able to monitor this wastewater at the airport throughout the pandemic, and we're able to detect viral loads throughout the pandemic. These trends in viral loads, they paralleled what we saw nationally, and even in some cases predated these national trends. When we had the emergence of new variants, we saw a rise in viral loads, and when we saw the emergence of variants at Schiphol Airport, they mirrored quite nicely what we saw nationally in the clinical surveillance. So what does this mean? This means that with actually very few samples taken at a single location, we already see quite a lot of trends we would see using a lot of more intense sampling. And that really makes that performing this wastewater-based epidemiology at just a single large international airport can already be a hugely valuable addition to classical COVID-19 surveillance. And I mean, imagine if we could do this in international context, in multiple airports in Europe or worldwide, then the potential would be much greater. Now, of course, this is absolutely not my idea. There are a lot of initiatives on this. There's a EU sewage sentinel system, and there's currently work on a global wastewater surveillance program, and hopefully this expertise that that's being developed might help us in tackling the next pathogen X, and maybe we'll have to use different considerations. So maybe not at the airport level, maybe at the airplane level, but that will be we'll see that when we get there, I think. So we presented this work first in Darwin at the Water Micro 23 conference, where we won the poster prize. I have to mention that, of course. And this work isn't just done by two PhD students. There's a massive consortium and an effort by a lot of people. So I have to mention our partners, Schiphol Airport Services, who gave us the passenger data. Of course, Avidus Industry Water, who collects the samples, sends them to us, of course, gives us information about what kind of wastewater treatment is going on. The research was funded by the Ministry of Health, and we have a large consortium that's working daily to process these samples and get this knowledge. And I've put some links for the preprint and for the data we used and the code we used in this research. Thank you very much.
Thank you so much. That was a really interesting talk. We have a couple of questions in the Q&A and then also some from the panelists. So we'll just get started with the Q&A. Were there any barriers that you faced in collecting and sharing this airport surveillance data? Were people concerned about the implications? Did you have any issues with any of that, or?
Well, so I have to say that I joined the program in 2021, at which point there was already a lot of sampling going on and information sharing. So for the complete picture, I would have to refer you to my excellent supervisor, Willemijn Lodder, who supervises all of this. But in my opinion, people have been very motivated to help. People are really interested. Schiphol Airport, the airport was really forthcoming with giving information on passenger counts. Our partners at Avidus Industry Water have given us a lot of interesting information and were really helpful. So that went really well. But of course, this is at the wastewater treatment plant level, which of course is less invasive than doing it at a single airplane, for example.
And then I have a little bit of a follow-up question on that. So what is the turnaround time from collecting the airport wastewater sample to actually getting the sequencing results? Like beyond just this like interesting connection, are they Like, did the government use this data? Was it produced in time to look at variants?
Yeah,
what's the turnaround?
So I think it was what we're doing is around 2 weeks. So we have the data, we collect the sample, the samples collected, and we have the data within 2 weeks. Sometimes earlier, we try to get this information as quickly as possible. We also do the sequencing is done in-house to just speed everything up, of course. And this data, specifically for Schiphol Airport, but also for the other wastewater treatment plants we have in the Netherlands, it's shared with, of course, the ministry and with relevant organizations. And we also have sharing information in international context just to get the information out there, because what's really important about tackling such a pandemic is rapid information gathering and dissemination, of course.
Mm-hmm. And then I had another question. So there was a really interesting association between increased viral loads whenever a new variant emerged. And I'm wondering if you have any thoughts if that is due to like the rapid spread of people getting sick, or if there's actually higher shedding, because it did appear that it was like exponentially higher for each new variant. So I'm wondering if you have any thoughts on whether it's just because more people are sick, or if it's an increase in the shedding rate of these viruses.
Yeah, so I'd have to say that I'm less well known with the immune concept of it all. But you could imagine that when a new variant is introduced, it's able to infect maybe new hosts or reinfect different hosts, and that could lead to more people being infected by this really distinct variant. Of course, increased shedding can occur, but not. But I don't expect it to occur in this fashion. So what we saw, I'll go a few slides back here. This is really a much larger shedding, and if it's a twofold increase. I would not expect that. No.
Okay. And then, are there any plans to sort of implement this surveillance at the airport for other potentially high impact pandemic diseases like Ebola, or is, like, because you have so many people traveling from around the world, it could be an interesting way to see how some of these more high impact things are moving? Or, like, are there plans to expand the program to other targets?
Yeah, so I would have to say I'm not I'm not involved at all the higher planning. So I know what's currently so we've had the COVID pandemic. We're still looking at SARS-CoV-2, and the expertise that we've developed, it can be applied to different pathogens. But of course, we also have to be able to compare our results and share this information. So what, to my knowledge, currently there are a lot of initiatives to combine this information, to work together, whether it's already specific for different pathogens. It may be in the works, but I think it would be a really good question for the EU 4S town hall, which is in I think, because I think it's gonna be a really big subject of that.
Yeah. Especially, it's an interesting sample because the treatment plant is only the airport and the companies on the ground. So it's a very different type of sample. It would be an interesting way to look at sort of how things move. And then we have another question about what are the plans for the future of this program? And then it seems like there is a lot of support for surveillance at the airport from the Dutch government, but will it continue? Are they going to keep up with this really intense sequencing program, or are there any plans to sort of dial it back at all, or anything like that?
Yeah, so the level we have previously done, of course, was really intensively for SARS-CoV-2. Of course, you can't do such an effort indefinitely because it's quite expensive. So what's now being done is there's being looked at what can we do, what kind of infrastructure do we need, and which other pathogens are available. So on sequencing level, there's also being looked at which pathogens are relevant, except for SARS-CoV-2. Now we are currently sampling at a slightly lower frequency, but it's really important to of course, keep this infrastructure in place, because it's really difficult to set up.
Great. Well, without any more questions, I think we can end a little bit early today. Thank you so much to both, unless Ali has something to add, but I just want to say thank you to all of our really great presentations today. They were really, really interesting and really informative. And we can give everybody a little bit of their time back.
Thank you. Bye.